Hello friends, welcome back to the channel. Let's continue talking about microbiology and the BACE. So let's get right into it. So I'll get my highlighter out here first, and so we're going to highlight this part right here where it says microorganisms are central to biotechnology. That's the big idea that we're talking about here. No bugs, no biotech. Why? Because microbes are these tiny factories. They break down stuff in fermentation, like beer, wine. Let me get my color just right. So in fermentation, um, they give us ideas to um, do more research or test ideas, and that all important drug production, things like insulin. So that's what this chapter is really about. It's not just about biology, it's particularly about zooming in. Microbes are, and cells, which are like the living tools, culturing methods, so how we grow them, and aseptic techniques, so how we keep things clean so we don't ruin our experiments, and just kind of real world uses um, of biotechnology. Let's just put a box around this word aseptic, right? So that means without sepsis or without contamination. So let's not overcomplicate it. Every fancy biotech breakthrough starts with someone keeping a microbe alive, pure, and useful. So one of the things I'm going to try to do, I'll do it in this red color here. We're just going to do some like BACE tips, because the BACE is going to love questions like why microbes matter. And they may ask you questions like which of these products, which of the following products rely on microbes. And so we can think about the acronym MFA, right? So microbes for medicine, microbes for fermentation, and application. All right, and at any point, go ahead and feel free to pause the video if you need to. So let's come to this next part where we're talking about microbiology and cell biology. So cells are the basic unit of life. Let's just go ahead and highlight that. Everything alive is built from cells. There's two types of cells. Prokaryotic cells, and these are the ones where they don't have a nucleus, or the nucleus is kind of free floating, and eukaryotic cell, which do have a true nucleus. So pro means no, eu means true. So microorganisms, let's highlight this next part right here, include bacteria, fungi, protozoa, algae, and viruses. Just a quick note, viruses aren't true cells, but the test still puts them in that mix. There are these three domains of life, RK, which are like prokaryotes that live in extreme places, bacteria, which are prokaryotes, like kind of everywhere, and then eukaryotes, which are plants, animals, fungi, protists. So a kind of another mnemonic device we could use to remember the three domains of life are going to be a big egg. All right, and this picture kind of looks like a big egg. Um, and humans are this teeny tiny dot right here on this bigger uh, branches. And most of life is just microbial. So common traps on the base. Don't mix up prokaryote versus eukaryote. Look out for questions like, which of the following has no nucleus? And another trap is there are no viruses on this domains of life. So let's come up here. Let's talk about history and Koch's postulates. So let's highlight that microbes have influenced human history, particularly through diseases like malaria, anthrax, and the plague. Um, Koch's postulates is going to be a recipe for proving that a germ causes a disease. So let's just underline that. So, couple of things is about Koch's postulates are that the bug must be found in every sick case, that bug must be grown alone in a pure culture, so it's just, must be found in abundance in all organisms suffering from the disease, must be isolated and then grown in a pure culture. That bug must make a healthy subject sick, so organism should cause a disease when introduced into a healthy organism. And you must be able to re-isolate that bug and it should show up again. So the microorganism must be re-isolated from the inoculated and be identical. So let's highlight right here. The next thing is that antibiotics like penicillin are part of that medical breakthrough. Um, Penicillin was the first true germ killer used in people, but because of over prescribing of antibiotics, we now have antibiotic resistance, particularly in cases like MRSA. So germs evolve and the drugs stop working. So a couple of base kind of tips, on the test, it may ask you something about Koch's postulates, maybe to put them in order, or it'll say which step is part of Koch's postulates. Um, Make sure you don't confuse antibiotics with antivirals. With antivirals. And remember, bacterial resistance is going to spread from hospitals. All right, so coming down here, looking at the structure of DNA, right? So DNA, or sorry, not DNA, of bacteria. And on here, the nucleoid, that's going to be where DNA hangs out. Plasmids are these extra little DNA rings. Ribosomes are how protein is made. And that cell wall is going to be made up of something called peptidoglycan. So let's make sure let's just box that word peptidoglycan because that's going to come up a couple times again. And there's some other things that'll have like flagella for movement and capsules for protection. So bacteria come in three different shapes. They are going to be either uh, sphere shaped, rod shaped, or spiral. And the way we can remember that is through the mnemonic device CBS. Put one too many C's there. And spiral shape. All right. Other things, let's come down here. Let's talk about gram staining. So there's always going to be a picture of two different bacterial strains, and you have to identify whether they're gram positive, which is going to be the ones with the thicker walls, or that thick peptidoglycan layer, or gram negative, and these are going to have that thin peptidoglycan layer. Gram negative bacteria is usually harder to kill with antibiotics. So that's going to be gram staining. And so maybe I'll just put the colors on here, right? So this is going to be purple, and the gram negative, that's going to be red, pink in color. All right, bacteria are going to have some specific environmental needs. And so this one up here is talking about the oxygen needs of bacteria. So if they are obligate aerobes, they're going to be found towards the top of this test tube because they're going to want to be around oxygen. If they're obligate anaerobes, which they don't want to be around oxygen, they're going to be at the bottom. Facultative, they're okay with either. And those are really going to be the three kind of big ones. You also have temperature preferences for bacteria. Some bacteria are going to be called thermophiles, and these are going to like really high temperatures. These are heat lovers. You're going to have some that just like to hang out in around normal body temperature. So, which is about, you remember, regular human body temperature is about 37 degrees, so it's right here. And then you're going to have psychrophiles, which are going to be really into that cold temperature, so cold lovers. And then you have halophiles, which really prefer to be in a saline solution or in a high salty area. So on the base, if we come back up to talking about the gram staining, um, you're going to, it may ask questions like which strain, um, or which stain result, results in a thick wall, or it'll ask you questions like, hey, which shape is this? Right, and then just think back to CBS. or it may ask you a question about which bacteria grows best at human body temperature. Right? And that's going to be mesophiles. All right, so let's come over here. I think that the temperature was the last one down there. All right, so biotechnological applications of bacteria. So let's use the word or the mnemonic bugs, as in microbes or bacteria, makes, bites, so like things like food, and biologics, so things like humalin. So bugs make bites and biologics. So when we're talking about bites, we're talking about food, like things like yogurt, cheese, or they're recombinantly engineered to produce things like human insulin. So different ways to culture bacteria, all of these, if it's in solid media, those are all going to be agar based. Um, and it's going to be in plates or in slants. Plates are good for spreading colonies. Slants are good for storage. And deep tubes are good to study oxygen levels or oxygen requirement. So LB agar, this is going to be for general growth. And then selective media is going to be to let some bacteria grow. And then differential media, these are going to show like things like color changes. So let's just do kind of like a little device here, a mnemonic device. So selective, those stop some, right? They stop some things from growing. Differential is to show differences. And then you have things like liquid media. So these are going to be broths, and these are used for larger storage or making lots of cells and proteins. So some tests you might see, or some test questions, is going to be something like, which medium distinguishes microbes by color? So don't mix up selective versus differential. Um, if a test question has something about large scale production, think liquid media. All right, so let's come back to, or let's continue talking about antibiotic testing. Antibiotic testing, so let's highlight the disc diffusion test or the Kirby-Bauer. Um, and so these antibiotics are placed on a bacterial lawn, and you're looking for the zone of inhibition around each of these guys. So let's get the red. So this antibiotic killed a lot of the bacteria. This did not kill any, um, and this killed just a tiny amount. So the zone of inhibition shows where bacteria can't grow. The bigger the zone, more effective the antibiotic. So a test question might be which test or another type of test question, it might be they give you two of these and you have to compare the zone between one antibiotic versus another, which one is larger. So if the zone is small or missing, like in this one right here, that means that the bacteria are resistant to that antibiotic. All right, so um, some techniques here in working with our microbes. So these are going to be the key tools. So an inoculation loop, that is just going to be for transferring microbes. Bunsen burners for cleaning. Incubators are going to hold at a steady temperature. And then fermenters, these are going to be for those large scale growth. All right, so aseptic technique, this is going to be that technique without contamination, so it prevents contamination. And then the streak plate method, this is going to be a way to spread bacteria step by step and end up with single colonies. So you kind of work in these quadrants as you divide it, and you keep going through these quadrants until you get to the very end, and then you could get just a few, um, 1 to 2 colonies. So let's talk about some base tips here. So aseptic is going to be to prevent contamination. We kind of talked about that earlier. Make sure you don't confuse streak plate with spread plate. So let's just do does not equal. Right, spread plate is where you're looking for that long. And then each of these colonies here, that is going to be um, like a clone of one cell. All right, so ways to count bacteria, um, how many you have. So these are going to be through serial dilution. And so you do these to, their end goal is to be able to count colony forming units or CFUs. And so if you have too many bacteria, you dilute again and again until you get to a amount that you can count. Right, so you're taking 100 microliters each time away from it, and you keep diluting again. Usually, somewhere between 30 and 300 colonies is about a good number to be able to count. And another way to get a general idea of how much bacteria is in is going to be to use a spectrophotometer. The spectrophotometer is going to measure optical density, or OD, to estimate the concentration of cells. So the cloudier it is, the more bacteria that's in there. Right, it's a quick estimate um, to get to it. So let's write down here. So plates, that's going to give you an actual count, and a spec, short for spectrophotometer, that's just going to be a quick estimate. So test questions you might see is which method gives CFUs? And that's going to be the plate method in serial dilution. Or it might say which tool measures OD? And OD stands for optical density, and that's always going to be done through a spec. All right, so let's come up here looking at gram staining, gram negative versus gram positive. We discussed this earlier, but remember, if it's gram positive, this is going to have that thick wall or thick peptidoglycan layer. Gram negative, this is going to have the thinner layer. So don't mix these colors. There will definitely be one test question about gram negative versus gram positive. Metabolic assays, the word assay just means test. And so these are going to test how certain enzymes work. So in starch hydrolysis, it's looking to see is how starch is broken down, or does it break down starch? Colony morphology, that's just going to be shape, color, and the spread pattern of the bacterial cells. All right, so let's come down here. Let's talk about eukaryotic cells. So eukaryotic cells, these are going to be a little bit more complex. They're going to have a nucleus, an endoplasmic reticulum, the Golgi apparatus, which packages things. Mitochondria is going to be ATP. Lysosomes, which kind of are there to break down any waste. And then the cytoskeleton, which kind of makes up the outer shell. And then plasma membrane. So if you are kind of confused as to what is in a eukaryotic cell, let's come up with another mnemonic device. So never eat green moldy lettuce. comma, cell poison. Like, oh my god, don't eat green some moldy lettuce. Right, so nucleus, endoplasmic reticulum, Golgi apparatus, mitochondria, let's capitalize L here, for lysosomes, cytoskeleton, and then you have that plasma membrane. Plant cells, these are going to have cell walls, you're going to have more shape, chloroplasts, so for photosynthesis, and large vacuoles for holding in water. Other types of eukaryotic cells, so yeast and fungi, um, those are going to be things that are used in like enzyme production or in making proteins. So, how are eukaryotic cells used in biotechnology? So, fermentation, think about bread, yeast for bread or alcohol, biofuels. And some of these we can do some recombinant work to make proteins. And the way you can kind of sort them is by the cheapest, which is going to be yeast, insects, which are the second cheapest, and then mammalian cells, which are the most expensive and like the hardest, but they're also going to be the most accurate. Um, so these are used for things like Herceptin in when we do genetic profiling for medicine. So stem cells, they can be embryonic stem cells, so from embryo. Um, these are pluripotent, which means they can be any cell type. There's also some kind of controversy with that. You can have adult stem cells, which are multipotent, but these are more limited use. And you could have induced pluripotent stem cells, or ISPC, um, which are adult cells reprogrammed back to being pluripotent. All right, so eukaryotic culture types, so primary cells, these are going to have limited growth because they eventually die. Immortalized cells live forever. So these are examples of these are going to be HeLa cells or Chinese hamster ovary cells. And a couple of ways you can kind of culture them is in adherent vessels. So these are going to stick to the sides of the wall, or in a suspension culture, which they're kind of just suspended um, in a large vat. Sometimes for with the growth media, they can use pH indicators like phenol red, which are going to change color if the pH were to change. All right, and then ways to store these things, right? So safety cabinets, like biological safety cabinets. These are going to protect samples from contamination. And then a couple ways to visualize them. So inverted microscopes, these look from below. Fluorescent microscopes, which are they tag molecules with like a glowing substance. or we could just stain them. One of the examples is going to be Janus green, or which is a green stain for mitochondria. All right, so that is kind of a quick rundown about the big microbiology concepts that you should be familiar with for the base. Um, Remember, just go back and look through the notes, look through these notes, and good luck and look out for the next video.